Siguiente comunicación. The role of myeloid-derived suppressor cells in enhancing HIV infection after a primary genital tract infection.
Okay. Good afternoon, everyone. My name is Dan Pira. I work by the Brown Institute de Recerca. And today I will present our project on the role of myeloid-derived suppressor cells during HIV, enhanced HIV infection. And what we know from literature is that sexually transmitted infections, or STIs, such as chlamydia, may enhance HIV infection in the cervical mucosa. And in this project, we question which cell type, immunological cell type, is responsible for this enhanced HIV infection that has been observed. And one of the cell types we were interested in are myeloid-derived suppressor cells, or MDSCs, which are immature myeloid cells that are activated as a consequence of pathology. And what is known from these cells is that they are potent immune suppressive cells that can suppress other immune cells. And so what we hypothesize is that in the case of a primary STI, such as chlamydia, the chlamydia infects the cervical mucosa, and these cervical epithelial cells start secreting cytokines, such as growth factors, MCSF, GM-CSF, and inflammatory cytokines, IL-6, IL-1 beta, and also anti-inflammatory cytokines such as TGF-beta. And these cytokines in turn attract cells that are needed to clear out the chlamydia infection, such as T cells, DCs, and NK cells. But the same cytokines, from these cytokines, it's known that they also attract MDSC cells and expands the MDSC population. So what we now hypothesize in this project is that in the case of the presence of HIV in the cervical mucosa, the expansion of MDSCs due to the primary STI may then also suppress the protective cells that are needed to fight off the HIV, leading to enhanced HIV infection and also replication. So what we had access to is that we included patients with a suspected STI, and from these patients we acquired PBMC samples, plasma samples, a cytobrush sample, and also a cervical lavage sample. In total, these were 77 patients, which were then assigned to certain groups, those that turn out not to have an STI, those that were diagnosed with chlamydia infection, bacterial vaginosis infection, HPV infection, or a co-infection of these pathogens. And in these samples, we performed flow cytometry analyses and also cytokine analyses. And in addition to that, we also had the chance to use cervical tissue of patients undergoing cervical surgery. And with this cervical tissue, we explored in the lab ex vivo if we can see enhanced HIV infection, which I'll come back to later. So then to the results, what we intended to do is to look at the myeloid-derived suppressor cells, and we used two markers for this, CD33 and CD11b, which you can see here in this pre-gate. And then within this orange gate, we look at two subsets of myeloid MDSCs, which are polymorphonuclear, or PMM MDSCs, and monocytic MMDSCs. As you can see here, we distinguish them by expression of HLA-DR and CD14. And in red are the MMDSCs, and what we see is that mainly there's a trend towards higher levels of these cells in the bacterial vaginosis group compared to the healthy control. And if we then look at the other subset, the PMN MDSCs, we see that it's a different picture. So we see that the chlamydia patients have higher levels of these cells compared to the healthy control group. So there seems to be a difference in the expansion of types of myeloid-derived suppressor cells by which infection there is present. We then also looked into the cytokine levels in cervical lavage samples, and we looked into growth factors, MCSF, GM-CSF, and VEGF-A. And what we mainly see is that the bacterial vaginosis group has high levels of all three of these growth factors. Only in the case of MCSF, we see in the chlamydia group that they are significantly higher compared to the healthy control. We also looked at inflammatory cytokines, IL-6, we do not see a difference there. For IL-1 beta, we do again see a difference in the bacterial vaginosis group, so higher levels, and also in the co-infection group. And as last, we looked at anti-inflammatory cytokine TGF-beta, which was also higher in the bacterial vaginosis group. Then I move over to the more ex vivo infection part, where we use cervical tissue, and we expose the tissue to elementary bodies of chlamydia, which are the infectious particles of chlamydia, for 24 hours. And what we then see is that in the case when we expose the tissue to the infectious particles of chlamydia, we see that there's an expansion of these HLA-DR negative, CD14 negative cells, which are the PMN MDSCs, which we saw in the patients that this population was also expanding. So what we see in the patients, we can replicate ex vivo in tissue. And this was in the individual patients, we saw a trend towards higher levels of these PMN MDSCs. And then my last results slide, we look into whether HIV infection is actually enhanced after a primary chlamydia infection. So what we did is, again, we had the cervical tissue, we treat the tissue with the infectious particles of chlamydia or with a cocktail of GM-CSF and IL-6, which is known to expand the MDSC population. And then this is for 24 hours, and then we expose the tissue to HIV for 10 days, and then look at the HIV positive or the infected CD4 cells. So what we see here is the percentage of P24 infected, so HIV infected CD4 T cells. We see in the case of no HIV infection that there's hardly any infection. If we do not treat with with chlamydia or the cytokines, we see that there's HIV infection. If we then first give GM-CSF with IL-6, there seems to be, and then HIV infection, there seems to be a bit higher level of infected cells. And we see a bit of the same trend for chlamydia with HIV. And if we then look into the individual patients, we indeed see that if we do not treat the tissue with chlamydia or GM-CSF, only with HIV, then we see an increase of HIV infection after treatment with GM-CSF and IL-6. And we see the same happening for chlamydia. So if we first expose the tissue to chlamydia and after HIV, we see enhanced HIV infection. So that leads me to conclude. So we see that different subsets of MDSC population expands based on the infection that's ongoing. And we also can see that ex vivo exposure to chlamydia enhances the HIV infection ex vivo. And our idea now is that different types of MDSC subsets that are induced by a primary STI can lead to different levels of suppression, and that's also that leads to a different or enhanced HIV infection. So what we intend to do is we still have the PBMC samples to be analyzed, and also we're moving to suppression assays with cervical MDSCs. That leads me to thank, of course, the patients that are participating in this study, my team at BIR, and also our colleagues at the Vall d'Hebron Hospital and Vall d'Hebron Drassanes. Thank you for your attention.
Muchas gracias. ¿Alguna pregunta? No. Muchas gracias.